Good afternoon, everyone. While everybody's trickling here, we're going to go ahead and get started. I'd like to welcome everyone to the Wild Neighbor Speaker Series. We're very happy to have you here today. This webinar is a series that's collaboration between Travis County Natural Resources and the City of Austin, who co-manages the Balcones Canyonlands Preserve, along with a number of other private and public partners. Before we get started, I want to introduce myself. My name is Jeremy Hull. I'm the community liaison for Travis County Natural Resources. Today, we are very excited to have Lisa Markovchick here to talk about Mycorrhizae101. Lisa earned her PhD at Northern Arizona University, where she is now a postdoctoral researcher studying the interactions between land management concerns and mycorrhizal fungi. Prior to this, she served as a DoD civilian for a decade, managing natural resources for the Navy, and received her master's from San Diego State University in plant and fire ecology. Lisa has created, developed, and led large restoration, management, and community science projects for over 20 years. And just recently, she spearheaded the first cooperative and community-powered mycorrhizae collection ever here on the BCP over the summer. Throughout this presentation and at the end, Jaya Torres with the City of Austin will be managing our Q&A sessions. So please feel free to put any questions you have throughout the presentation in the Q&A box, not the chat, and we will get to as many of those as possible throughout the presentation. There will also be a recording of this made and posted to our Facebooks and YouTube channels to be used as a future resource. So on that note, I'm going to stop sharing my screen and hand it over to Lisa.
Thanks, Jeremy. I'm going to go ahead and try and share my screen here. Hopefully you will be able to see it. Can you see it okay?
Looks great.
Awesome. All right. So thanks for joining me, everyone, and joining us here today. I'm going to talk about Mycorrhizae101, which is sort of what Jeremy had asked for. He thought that might be really interesting for folks. And I'm going to cover sort of 3 different sections here today, and we're going to have little question breaks sprinkled throughout. So if you have a question that comes up, feel free to enter it in the Q&A. And then that way, when we get to the question section, we'll have those available right away, and we can take some of those. So first, I just wanted to talk to you guys about what mycorrhizae are. And then in the second sort of area, we'll talk a little bit more about what they actually do in our ecosystems, because I think that's really important and not broadly known. And also we'll talk a little bit about how are they doing conservation-wise, what do we know about that? And then at the end, I thought it would be nice to just kind of end by talking a little bit more about what we're doing at the BCP. So let's go ahead and talk about what are mycorrhizae. The term comes from the Greek mykes, which is just fungus, and rhiza for root. And so you can see there, it's literally a fungus root. And you've probably seen pictures like this before, where you can kind of see on this little pine seedling, the brown roots coming off, and then sort of radiating out from those, the white hyphae, and those hyphae are actually connected to what we would call an ectomycorrhizal root tip. So we're going to talk a little bit more about all of that. And they were first described as a mutualism or a symbiosis sometime around the year 1880. The references that you can find kind of differ a little bit, but by this gentleman called Francisc Kamienski, hopefully I didn't butcher his name, with some drawings here that you can see, those ectomycorrhizal root tips. And they are just an ancient symbiosis that originated sometime between 500 million years ago and 130 million years ago, depending on which one you're talking about. Nearly all land plants depend on them, so about 85 to 90% of land plants. And the fungus part receives plant photosynthate for the resources and services that they provide to the plant. There are several different kinds of mycorrhizae based on the plants that they partner with and also the structures that they form when they sort of form that interface with the plant. So I've shown you 2 here, Arbuscular or AMF, named for that little thing that looks like a tree. It's called an arbuscule. which is actually the interchange between the plant and the fungus. And then ectomycorrhizae, or what we would call EMF, named for this sort of little sac swollen root tip that you can see in that picture there, and just very much known for helping plants. So I'm going to talk about those first two kinds first, since they're sort of the dominant ones in a lot of our ecosystems, and then we'll go through some of the other groupings that are a little more rare. But let's start with AMF. They are kind of the original ones. They're the guys that helped plants move to land. That's that 500 million years ago evolutionary event. They all came from this one kind of evolutionary event. And I like to call them roots before roots existed, because when plants first moved to land, they kind of didn't really have very good root structures. So these guys sort of helped them by helping to form that root structure. They're all members of the Mycorrhizae, which is just a grouping taxonomically, and as a result, they're taxonomically less diverse. There's about 200 of them described. They tend to partner more with the herbaceous and crop plants, so we know them a lot from agricultural studies, just because there's sort of in the modern era, of course, been a little more research going into the agricultural industry. So we have some good studies from agriculture talking about these guys, but they also do actually partner with some woody trees like juniper, and they're typically less visible. So that formation of the arbuscule occurs inside the root cells, and when they reproduce, they form underground reproductive organs and spores, we think, because we often can't see them, and in some cases we've actually never been able to find spores. So there's actually a question as to whether all of them sexually reproduce or not. And then on the right here, you can see these little oval or round shaped nodules, and those are called vesicles. They're thought to be a storage structure also inside the root cell. Ectomycorrhizal fungi first appeared about 130 million years ago. They are more taxonomically diverse, primarily because they have evolved independently at least 60 times that we know of. So this is actually really cool, because they sort of tended to start out as something that we would call an alternate lifestyle. So for instance, they might be a decomposer fungus. And then they became aware that they could kind of form this symbiosis with plants. And so a lot of cases, not only does that mean they're more taxonomically diverse, but they can also be kind of more specialist, because they really evolved a relationship with a certain plant species or plant genus. They tend to partner more with woody perennials, so think things like pine trees, but some plants like cottonwoods and oaks actually have both AMF and EMF. They also tend to be more visible, so if you're a mushroom aficionado, these would be the guys, in addition to decomposers, that you're probably more familiar with, because they form mushrooms, and then they also form this structure that's actually on the outside of the root tip. So we can see them a little better and more easily just under a dissecting scope, and sometimes even with our bare eyes, depending. And their interchange structure between the plant and the fungus is called a Hartig net. So you can see that in this picture here on the top left. The white portions are the plant root cells, and then that sort of net-looking thing that's kind of dyed sort of a bluish gray color is the Hartig net itself. It's actually the fungus incorporating itself amongst all of those root cells on the sort of outer edges of those root tips. And I just wanted to show you what that all kind of looks like taxonomically over time. So along the bottom here, we have millions of years ago, and we can see as the fungi start to emerge taxonomically in the yellow box, and then within that, the Glomeromycota or the AMF emerge first. And then even further down along the line, we have several different groupings that all sort of have EMF included within them, because the EMF, keep in mind, have evolved multiple times throughout evolutionary history. And again, you can kind of see a similar trend through time here, but I just wanted to show you what that looks like with how our plants are actually forming. So the AMF crop up first with these plants that are just moving to land who don't really have very good root structures. And then as we move along the line, once conifers become and gymnosperms become more dominant, the EMFs start to crop up in conjunction with them. It's actually really hard to find out this information. Certain scientists have worked really hard because, of course, mycorrhizae are all made of soft tissue. So if they don't have bones, they don't have teeth, they don't really have things that preserve very well. However, there are some really cool studies where they have actual photos of some of these fossils, and I just wanted to show you them because they're really cool. So this is what an AMF from 400 million years ago. And again, you can actually see that root cell with the arbuscule, that little tree formation inside the root cell. That's the interchange between plant and fungus. And here's one from the Middle Eocene showing you an EMF. So on the left, they're showing you that root tip, like a cross section of that root tip that you can see is kind of swollen. And then on the right, they're actually showing you again a picture of that hard net, which is the fungal structure being incorporated into that root tip cells. And there was a really cool study from Randy Swati, who actually works for the Nature Conservancy now. He did this study in 2016 in conjunction with one of my advisors, where they looked at the different plant associations across the US. And based on what we know about which types of fungi associate with which types of plants, they rendered sort of, a model of what the mycorrhizal associations across the US looked like before European settlement. And so the yellow ones are AMF dominated and the purple ones are EMF dominated. But like I said, there's several other groupings. So I just wanted to kind of touch on those a little bit and make you aware of them. They tend to be smaller groupings because they're really specialized with regard to the plants that they're associating with. So for instance, ericoid mycorrhizae are from the plants are from the Ericaceae, and they're usually in heathlands. This includes things like rhododendrons and cranberries. The fungi have these intracellular hyphae in the roots, and they also act as decomposers. So interestingly, they're actually both at the same time, where they're decomposing things and then they're transferring those nutrients to the plants. So these guys are thought to be very important in these sort of nutrient poor systems. There's also monotropoid mycorrhizae, which some of you may have probably seen, for instance, like pine drops. The plants are from the Ericales in the Monotropaceae, and the plants themselves have no chlorophyll. So they're actually mycoheterotrophs, which just means they're getting their energy from the fungus. And then the fungi form an ectomycorrhizae with another plant. Interestingly, again, like I said, it's not just based on the plant, but also they seem to have these different structures. So in this case, they would form what's called a hyphal peg, just pictured here on the bottom left. So different plants and different structures. And then orchids actually have a whole grouping of mycorrhizae because they're really heavily dependent on their mycorrhizae. The plants are from the Orchidaceae, and most are photosynthetic once they reach adulthood, but some actually never are. The fungi have also alternate lifestyles again. So they may be decomposers, or they may form other types of mycorrhizae with other types of plants. But in orchids, they form these coils of hyphae inside the root cells, and those are called pelotons. And there was a discussion for a long time about whether these orchid mycorrhizal fungi are really a mutualism or a symbiosis, because since the plants go for so long without photosynthesizing, are the plant and the fungi both benefiting? Because orchids can be as old as 11 years old before they photosynthesize for the first time. And so they have sort of this extended period where they're not photosynthesizing, and people wondered whether the fungi were really benefiting. And evidence suggests that yes, in fact, it is actually a mutualism. It's just that there's delayed reciprocity. So it's kind of like having a credit card, right? They're charging up a whole bunch of services from the plant, and then once they actually, from the fungi, and then once the orchids become photosynthetic, then the fungi benefits once the orchid becomes mature. So kind of a cool system. And something that I think is really important to point out is we talk about these mycorrhizae in these large groupings, but in actual fact, there are, for instance, in this case study, this one orchid species had 328 different taxa or species of fungi associated with it. And so, of course, that's an extreme example. Orchids are very heavily dependent on their mycorrhizal fungi. Not, it wouldn't be that high for all plants by any means, but I just use that example to point out that while we're talking about them in these big groupings, really that diversity within that is actually very important. And so that's kind of just what mycorrhizae are. And I can go ahead and take some questions for a few minutes if there are some before we go on to our next section. Let me know what you think.
We do have a couple questions. First question, is there any known sexual reproduction in Glomeromycota? I think I said that right.
That would be the AMF. So yes, there is some sexual reproduction where people have found spores, but that's not necessarily true for all species or genuses.
Okay, and then the next one, this person says, I just planted some Central Texas natives in my garden. Is there a way to introduce some of these EMF? Is there a way to test and find out if these fungus have connected with my trees?
Yeah, so that is a great question. We're going to talk a little bit more about some of what we're doing at the BCP at the end, which I think will help sort of clarify that. And I think that's probably the best I can do for now is we'll talk about what we're doing at the BCP at the end. And then our goal is to kind of have an ongoing program with the community at the BCP. So hopefully, maybe they can become a source of mycorrhizal fungi for your gardens, for instance.
All right, great, thank you. And then someone asked, can we get these slides sent out for reference? We are recording this webinar and we'll post it on our YouTube channel and Facebook pages, if that's helpful. All right, and that's all we've got in the Q&A for right now, so we can go ahead and go on.
Okay, sounds good. So yeah, you guys are going to post the YouTube results, and there are some really cool illustrations that we've had done here. So I just want to ask that folks not reproduce those because they haven't been published yet. It just takes forever to get things published. So as we go through here, hopefully those illustrations will be helpful, but I'm just going to ask you to, out of respect for the publication process, to not reproduce them. So I thought it'd be really helpful to talk about ecosystem services provided by mycorrhizae. And keep in mind that mycorrhizae, the term is actually plants and fungus, right? Fungus root. And sort of what I'm talking about predominantly here is what the fungi are bringing to the table. So on the left, we have a whole bunch, sort of a depiction of a whole bunch of ecosystem services that mycorrhizae provide. And on the right, just a depiction of what, the lack of those ecosystem services would look like. And we're going to kind of go through examples of a whole bunch of them here. So the first one is always that mycorrhizae do mine nutrients from the soil that would otherwise be inaccessible to plants. And then they provide them to the plants, which in turn improves plant health. And just like you and me, if the plants are healthier, they're more resilient to all kinds of stressors like pathogens, disease, drought, all those different kinds of things. And that's pretty well established even just in the agricultural literature as well, because mycorrhizae are known for making food more nutritious as well as a result. And this is actually super important under climate change, and not something that maybe we all think of all the time, but climate change is expected to make food less nutritious. And so mycorrhizae are actually seen as one of the ways that we can help to counteract that by giving our plants, our crop plants, the mycorrhizae that they need. Sorry, I always forget how to move slides along in Zoom. And the other cool thing that mycorrhizae do that's especially important these days is they help sequester carbon. So because they're providing all these nutrients to the plants, they're actually boosting the carbon that's being sequestered in the plants themselves. And then they're also, remember, they have tissues in the soil that are sequestering carbon, in addition to this glomalin that they secrete, where that is actually storing carbon in the soil itself as it aggregates and holds that soil together. They also do things like change flowering windows. So this was a completely surprising one that I found as I was looking through all the ecosystem services that they provide, where they're actually known to decrease the amount of time before a plant can flower, and they extend that flowering window. And again, this is thought that to be very important, particularly under climate change, because with the weather changes that we're seeing and more extreme weather and so on, there's a significant chance that plants might be flowering before or after pollinators are there, and that there could be a timing mismatch. And so the effect of mycorrhizae to go ahead and extend these flowering windows could be really important in ensuring that plants and pollinators can get their timing right. And common mycorrhizal networks, or CMNs, are known to transfer carbon not just between plants of the same species, but also between plants of different species, which is pretty cool. And these common mycorrhizal networks are also known to assist with pest control. So they actually can carry signals to warn neighboring plants, for instance, in the study of an aphid attack. So the plants that are not yet under attack sort of have an advance warning and can send out phytochemicals in their leaves that would make them less attractive to the insects. They also allow the plants to go ahead and coordinate the chemical signals that they're sending out, so such that they can rally parasites and predators of the insects that are attacking them. So for instance, in this study, they were able to send out their volatile chemicals together, so that makes for a stronger signal to attract these wasps that are actually parasitic on the aphids that were attacking them. And if I forgot to say what a common mycorrhizal network is, it's just the fungus linking up the plants. The thing that I get really passionate about, which I think doesn't get talked about as much, so we had this illustration made just so we could talk about it, is that mycorrhizae also provide a whole bunch of water services to the local ecosystem. And I'm trying to move my little bar around so you can see it, because I don't know if it's in your way. So we're going to take a look at each of these, but just to give you an idea overall, they help plants modulate their water use efficiency by modulating their stomatal openings. They help move water vertically and horizontally. They're also helping to change the way that water infiltrates and is retained in the soil because of the effects they have on the soil structure. And as part of all of that, they're part of this local water cycle and evaporative cooling cycle. So just a few examples for you. For instance, in this study, the addition of mycorrhizae promoted survival and growth in a drought, so helping to modulate that water use efficiency through stomatal openings. They were actually able to increase shoot biomass, increase survival, and increase shoot water content during a drought. One of my advisors here at Northern Arizona University did this amazing study on pinyon pines. So in the picture, you can see there's actually two genetic strains of pinyon pines. There's one on the left that's kind of taller, and one on the right that's more stunted looking and shorter. And you can see under a drought what their relative mortality was. So on the right, we have a drought-tolerant genetic strain, and on the left, a less drought-tolerant strain. And she was able to show that they actually have very different mycorrhizal communities as well. And the really interesting thing was, if you took their offspring and looked at what mycorrhizae they would associate with, that community held the same. Even if you tried to inoculate them with the other mycorrhizal community, they would sort of accrue the mycorrhizal community of their parents. And even more interesting, she was able to show that under drought conditions in a greenhouse, they actually tried the ones that are sort of less drought tolerant, so those ones on the left, actually tried to acquire under drought conditions, started to try and acquire the mycorrhizal community of the ones that were more drought tolerant, the ones on the right. So pretty cool studies that have been done with regard to drought. And then also water infiltration in the soil. So because mycorrhizae are secreting things like glomalin, which are aggregating soil and holding it together, it's kind of like a glue almost. They're affecting the pore sizes in the soil particle sizes, and how the soil is clumping. And as a result, they're actually increasing that soil water availability to plants under drought conditions. So this is just a study where they showed, for instance, that decreased hyphal density or activity resulted in decreased soil moisture. So also, what you're seeing here is water in mycorrhizae that's been dyed with a fluorescent dye. So at the top, you're seeing that in ectomycorrhizal hyphae, then below that, arbuscular mycorrhizal hyphae, and then on the bottom there, the root EMF interface. And so plants on a nightly basis will lift water from the lower soil layers where it's moister into the upper soil layers where it's drier, so that it will be more available to them during the day. And this is called hydraulic lift. And in this study, they were able to show that both AMF and EMF helped conduct this hydraulic lift during a drought. And that was in partnership with sage and oak. And there have been studies kind of using similar measures, either stable isotopes and or this fluorescent dye, where we can actually see that there is some horizontal sharing among plants of water during drought. And that can happen sort of two different ways. You know, usually people study how it's being transported through the common mycorrhizal network, just like the previous slide we were showing. But also, interestingly, in the picture below this illustration here, you're seeing a hyphal tip that's actually leaking water out into those upper soil layers. And so even if you're not on that common mycorrhizal network, there's a chance that you could actually be getting some water as a result. So that little call out bubble there in the illustration is just trying to show those two different methods where it could be on the common mycorrhizal network and you're getting water that way as a plant, or even if you're not hooked up, but the hyphae are leaking it out into those upper soil layers, that could be helping you. And there's some really cool studies on this. I'm just kind of racing through some examples. But for instance, in this one, the researchers fed water to the trees in the little red bubbles. And then they had this diagram of where everything is and how the water spread. And so you can see in sort of the dark blue, there were trees, little seedlings that those trees shared the water with via common mycorrhizal networks. And then there are, by day 15 and 21, there are these other trees where water was also shared with them, and it came not only through the common mycorrhizal networks, but also via the roots in the soil, kind of like we were just talking about. Of course, the next question was, what do we know about how they're doing, right? And unfortunately, these guys are super hard to observe because they're underground and super cryptic and secretive, and we have to DNA sequence them just to find out who they are. And then we have to use things like, stable isotopes to find out what they're doing. So lots of different complex techniques are required, each of them having like multiple sets of specialists to bring them to completion. Also, unfortunately, there's a lot of uncertainty whether things besides plants and animals are even covered under the Endangered Species Act. So there's really not a lot of conservation listings either within the US, or even if you look at international listings, a lot of the time, like the IUCN Red List is trying to review fungi, and it's usually the mushrooms that we know and love and eat. and things besides that tend to just have data deficient as the resulting listing because we don't have enough information. And as a result, because they're not really protected, there's also kind of a lack of funding And even the funding that does go to fungi tends to go towards the ones that cause disease, which is super important, but that sort of leaves us not really doing a good job protecting and working with and implementing what we could with the beneficial fungi. So all of this kind of leaves us in what I like to call the dark ages of fungal knowledge compared to charismatic megafauna like plants and animals, because we're really still trying to figure out even what constitutes an individual species or taxa sometimes. And we're still trying to figure out like what are those specific functions that one species is doing that maybe other species aren't doing. What's their range and population size and survival and growth rates and all those things that we would need to actually know based on a particular species or taxa how they're doing. And we've been talking about them as big groupings, but this diversity actually really matters at the species and taxa level, because you can see again and again in a lot of the studies that I've been talking about that only some of the mycorrhizal taxa in a habitat perform a specific service in conjunction with a specific plant. And so even though we're talking about them as these big groupings, I just want to really point out that that diversity that we're still trying to learn about actually really matters. Unfortunately, the data that we do have from studies has been showing that many things actually do negatively impact mycorrhizae. And in many cases, it's the same things that maybe we would expect to impact them, you know, that are similar to what impact plants and animals, like land use change and habitat fragmentation and drought and things like that. But also it can be not just the invasive species of plants that are coming in, but even the herbicides and pesticides that we're applying to try and get rid of those, as well as things that we don't really see that much that maybe we don't think about, like the deposition of pollution from the air into the soil. And unfortunately, what we're learning from the literature is that ignoring this actually seems to be creating these self-reinforcing conditions such that even when we go and try to restore the above ground habitat, we're not really getting great results. And that seems to be related to the fact that we're ignoring what's going on below ground with our mycorrhizae. So for instance, to use the invasive species example, if we have invasive vegetation move in, that's reducing our native plant hosts. And that in turn is having impacts to reduce and shift our native specialist mycorrhizal communities underground. And then when we go to restore our native plants on the bottom left there, it's still impacting them. They're still not really growing the way that they should be after we've tried to restore the above ground, because we haven't addressed that below ground component. And this seems to be creating a continued advantage for things like invasive vegetation. Because maybe they don't need the mycorrhizae, for instance. And in some cases, there's even a study that shows that once your mycorrhizal community is shifted and depleted, if you're ignoring that and not addressing that, it's possible that the invasive vegetation is actually using the mycorrhizae that are left to steal carbon from the native plants. So really scary. And so we can see this in one of the studies that I've been working on after a tamarisk invasion in our riparian areas, just the soil legacy. Once the tamarisk is gone, just that soil legacy is reducing our cottonwood survival quite dramatically. The good news is, yay, good news, is that if we can actually restore our specialists and native mycorrhizal communities and their full diversity, this has really positive effects. So study after study is now showing that it reduces that weed biomass, it's increasing native plant survival and growth, and it has potential to increase suitable habitat for other species as well. And so just a few examples of that. For instance, this was a study done in the Midwest after agricultural disturbance. Keep in mind that agriculture plants use AMF. Prairie plants, herbaceous, also use AMF. Even so, they had to inoculate with a diverse mix of the native AMF community that was much different and more diverse than what would have been there after agriculture, in order to get some species to grow, period. And then the other species showed increased survival, increased productivity, and so on. And like I was saying about how that diversity matters, there's even a study showing that which mycorrhizal community you pick can actually steer what community or ecosystem you get after restoration. So in this one, they had an abandoned egg field, and they could either get a heathland or a grassland, depending on what mycorrhizal community they restored. So just a caution there that our plant and fungal and site pairings really, really matter. And you can actually get neutral to negative effects if you get that pairing wrong. So something that is actually totally worth it. We get huge results and the benefits increase with time if we restore our native mycorrhizal communities, but we just want to be really careful about how we do it. And that is what I had for that section. So we could probably go ahead and take a few questions before we talk about what's going on at the BCP with mycorrhizae.
Yes, absolutely. We've got a couple questions, so we'll take about 5 minutes. So I know you talked about invasive vegetation effects. Do you know anything about how invasive worms are affecting mycorrhizal communities?
That's a great question. And I don't, as far as I know, no one's looked at that. And I would be interested in hearing, I don't know if you guys have information on if you have invasive worms at the BCP. I haven't heard of that. But I know I've seen stories, of them around. So as far as I know, no one's looked at that, but I'd be interested in hearing what you guys know about if they're in the area.
I agree. It's an interesting question. And then there were a couple of questions about how prescribed burns on a landscape affect a mycorrhizal community.
There are some mushrooms that follow fire, and I'm sure that there are some studies. I mean, even just mushroom foragers will tell you, right? Following a fire, that might be where you want to go to find some morels, for instance. But there are studies, and that's a big question because every fire is so different, right? Because you have to think about fire severity and timing and all these other things. So I would say the literature's out there, but it's not like I can sort of give you a quick answer on what that interaction itself is.
Okay, thanks. And then how are mycorrhizae affected by soil temperature changes and soil pH values?
Soil chemistry of all kinds can affect mycorrhizae. So I am not thinking there's a single answer. For instance, as we talked about, some of those mycorrhizal types are found in very, very different communities, and it would probably be community dependent and even taxa dependent. But for instance, like one of the things that's thought to affect mycorrhizae after tamarisk moves in is, of course, the salinity, because the salinity does change. So again, there wouldn't be necessarily a single answer. This is where that diversity is really important and matters, even though we don't necessarily know every single taxa and how it's affected by one thing or another. It's going to vary.
All right, and then we got time for one more right now. Are commercially available mycorrhizal powders beneficial to add to the roots of plants?
This is where I have to say a strong, hard no. So there are multiple studies now showing that and not just commercial, but mass-produced inoculum is really not a good idea for several reasons. In some cases, it doesn't actually have live mycorrhizae in it, and it contains fertilizer instead. So there's that, but then there's also the fact that the diversity matters and the pairings matter that we just talked about. So anytime you're mass producing something, particularly since you probably don't know where it came from, it's not necessarily local, but it's also potentially, it could be invasive, right? We haven't really talked about fungi being invasive yet. But think about plants and when you move them around, what happens, and the fact that some of them become invasive. And then also the ones that are sort of mass produced are usually the ones that are easiest to culture. and package and things like that. And quite often mycorrhizae actually are not easy to culture because they are really dependent on this symbiotic relationship. And so for many, many reasons, including that point that we talked about where the pairings between the site, the plant, and the fungus really matter, and the diversity available within each of those. For all those reasons, the commercial inoculums is not something I could recommend.
And it's kind of on the same page. Is there ever a case of too much mycorrhizae, like when you're doing inoculations?
So too much, not necessarily, right? But again, it would come back to that pairing question. And then the question that other people always like to ask is like, is there a time where mycorrhizae are maybe not beneficial, which is sort of an extension of that question. And yeah, you can get different results with mycorrhizae if you put the plants and the mycorrhizae in strange situations. There's a spectrum of results that you can get. For the most part, it's a symbiosis and a mutualism where both parties seem to benefit. But are there occasions where maybe we don't see that in the short term, or maybe we could create an artificial situation where that's not true? Absolutely. For instance, if you add a lot of fertilizer to your plants, then they're going to need their mycorrhizae far less, right? Because part of what the mycorrhizae are doing are helping them to access nutrients in the soil that are not available to the plants themselves. So if you're sort of releasing them from that, then they might not need their mycorrhizae as much, and things like that. So hopefully that helps.
Great, thanks. And then this next question, and then it'll kind of move us into your next section, I think, is there any simple way to observe mycorrhizae in nature?
Yeah, so the sort of quick answer to that question, one is we talked about how ectomycorrhizae are the ones that form mushrooms, right? So those are ones where the mushroom is, of course, just sort of their fruiting period. But during that fruiting period, you can obviously observe them. And we're going to talk a little bit next about the root sampling that we did and things like that, and where we're hoping to take that. So maybe I'll go back to move right into what we're doing at the BCP. Can you see my screen?
Not right now.
Okay.
But yeah, we still have a couple of great questions, and we can save those for the next Q&A section.
Okay. Yeah, so let's talk about what we're doing at Balcones Canyonlands Preserve, because I think it's amazing. Hopefully some of you were there with us in September and got to take some root samples. So I just wanted to talk a little bit about what we're actually doing there. I'm moving my little Zoom bar around to try and make sure it's not not in front of other things. So basically at the BCP, we're trying to answer some of the basic questions that the literature would sort of steer us towards. So a lot of that literature that we talked about of the promise of mycorrhizae, if they're intact, and what that can do and how we might need to restore them. So sort of the primary question would be, where in the BCP are mycorrhizae more diverse and abundant? And could these locations then serve as inoculum sources for things like restoration projects? And also, based on the literature, right, we would expect that if we have a really good intact mycorrhizal community, that fungal diversity would then also be related to other things like plant diversity, habitat quality, plant resilience in drought, or other things. And so we want to see if we can find some of those patterns going on at the BCP, both in the positive and then also in the negative, where in the BCP maybe have the mycorrhizae been depleted and shifted because of, invasive species or other things going on. And if we can locate those places, then maybe could we restore them? And would that restoration help improve our plant resiliency when we have droughts and things like that? And part of the other whole point of the project, which I think is really cool, is to see and sort of show the world whether we can work with the community to do mycorrhizal studies. I've had lots of people tell me that it wasn't possible, which really kind of made me mad because I started out as a volunteer coordinator doing plant monitoring on Superfund, like after rehabilitating Superfund science with volunteers, and they were amazing. And the community, you know, is always so amazing and a wealth of resources. So I think we proved in September that yes, indeed, we can, for instance, collect these mycorrhizal roots with the community. And as we go along in the next few years, the next question is, you know, to what extent can we push that? Like, what are the additional things that we can actually be involving the community in doing? So such that, you know, if the BCP desires, they can sort of have this ongoing monitoring and adaptive management program where they're incorporating the mycorrhizal aspects into all of their other management. And just additional questions, because we have quite a few of them, like we sort of talked about the basics, but just to give you a few more specific examples is, for instance, we were looking at some of the places where we know we have invasive species that have come in and then perhaps also been treated. And we wanted to see, you know, are the different kinds of invasives having similar effects? So for instance, the exact effects on the mycorrhizal community can depend on what invasive plant we're talking about. So for instance, if we have vinca versus ligustrum species, are both of those sort of affecting mycorrhizal communities in a similar way or not? And are they even both affecting the mycorrhizal community, period? And then if they were treated, say with herbicide, since we know the herbicides and pesticides can also affect mycorrhizae, is that having an additional impact, or is the invasive species itself just, you know, having such a huge impact that the herbicide treatment, you know, it doesn't really matter at that point? And then one of the other questions that we're looking at, which I think are important, and interesting, is that Austin actually does this, BCP does this amazing job with shaded fuel breaks, which I think is like, they could be a great role model for a lot of other areas. And shaded fuel breaks are just the idea that, you know, where the BCP sort of, you know, butts up right against these neighborhoods, that we want to try and reduce some fuel based on the firefighter suggestions. But we also want to try to preserve our native plant diversity and canopy, both because we value it, but also because that native plant diversity and canopy actually also protects against factors that would you know, if we change them, would increase fire risk. So by keeping that native plant diversity and canopy, we're actually, you know, providing protection against increased wind speeds, which would make a fire worse if we didn't protect against them. And we're also keeping cooler temperatures by keeping that canopy and its evaporative cooling going on, and keeping sort of our humidity and our soil moisture. And so one of the questions that we have about mycorrhizae, since mycorrhizae are such a key sort of tool for the plants in keeping hydrated during a drought and things like that, is, are these shaded fill breaks also able to preserve the mycorrhizal fungal abundance and diversity? Because if so, then we could hold that up, right, as a really great best management practice for land managers. And we would have that knowledge as we move forward to manage the BCP. And if not, then maybe we could actually restore our mycorrhizal communities in those areas, which would, you know, again, help with plant hydration and things like that. And so there's a sort of a whole raft of questions like this that we're looking at. But I just wanted to give you a few sort of examples and dig into them a little bit. And what we're hoping is that as we go on, we'll start to be able to work with folks in the community to not just collect the root samples, but then also see what other aspects of the mycorrhizal sort of colonization counts and things like that can we get the community to help with. And, you know, also talk to the BCP about how they want to then use the results. Either, you know, as part of the management for the BCP and working with the community, say if folks have native plants, for instance, that they would love to have a mycorrhizal source for. And so I think that's about it. Before we go to questions, I just wanted to, say a big thanks to everyone, particularly Balcones Canyonlands Preserve, for just being willing to step up and actually, put their money where their mouth is and really become a role model for land managers, because not a lot of land managers are really doing mycorrhizal stuff yet. And this is pretty cutting edge. So Balcones Canyonlands Preserve is just doing an amazing job. And also to all the sponsors and people in the lab and people in the field, including probably folks on this call who have been helping with this work and making it possible in one way or another, because it just wouldn't be possible otherwise, both the research that I've been doing, as well as, even just my time to do presentations. And so here's my email address at NAU. And also I'm working with a nonprofit called Wild Earth Guardians. We're hoping to build a program as well to make sort of education opportunities around mycorrhizae and training opportunities for land managers more available as well. And so feel free to contact me if you're interested in collecting root samples and things like that. I'm pretty busy these days, but I will definitely try to always get back to folks when I can. So yeah, I think I'll probably stop there and ask if there are questions.
Yeah, we have a couple more minutes for some questions. Let's go ahead and start. Do you know if there are ways to artificially introduce glomalin into the soil, and would that be of any benefit?
I am not aware of any ways to artificially introduce it into the soil. I'm sure it's possible, but I guess my question would be, why wouldn't it make more sense to actually restore the mycorrhizae that would then go ahead and keep it in the system and in an ongoing way? Yeah, because really it's kind of all, it actually holds the soil particles together. So it's actually integrated with all the different soil particles. So I feel like doing that artificially would be a pretty tough lift, but also that restoring your mycorrhizal community would probably be better in the long run and do, have the same benefits.
So in addition to restoring mycorrhizal communities, how can we support myco health and determine whether an environment has a healthy myco community? If there are any ways to determine that.
Yeah, so there's, first of all, like the simple way, right? The simple way is think about the land use history that your area has undergone and some of the different things that we talked about. And there's a HART, what year is that? 2017 paper? HART that is focused on AMF, but they actually have it like a little flow chart where they say like, here are all the different disturbance types, and it probably has even gotten a longer list since then, but they kind of say also look at if you can't assess your actual mycorrhizal community because it's too expensive or whatever, think about that land use history and what has happened there. And you can kind of infer from that what the effects have probably been, because the results are pretty clear from the literature that, for instance, herbicides affect mycorrhizae, and pollution deposition and all these things that we talked about, invasive vegetation. So that's sort of the easy way to sort of infer. And that is something that we're actually just trying to get a paper out where we have like 3 action items and 10 tips on how to implement them for land managers, of things that you could do. That's like one of the first ones is when you're thinking about protecting and conserving, whether it's your private property or sort of natural areas on public land, that you could infer from the land use history what your status of mycorrhizae probably is. And then as far as doing the restoration, we're hoping to get some trainings going on how to do that, and sort of guidelines, because again, the diversity is really important and the pairings are really important. And so we're trying to find a way where we can sort of sort of set aside time to talk about that in depth and talk about the ethical and biodiversity considerations with folks so that everyone's not only educated in the methods of how to do it, but also educated in some of those concerns that we want to be thinking about as we're doing it. Like, we don't want to spread oak wilt, right? So those are some of the things that we're thinking about with this project, is also how do we do that and make sure we're not spreading oak wilt, for instance. Yeah, so it gets a little more complicated. It's not a complete answer. And we're trying to work on bringing sort of a fuller training and picture of what that looks like to folks.
Great. So aside from things like oak wilt, do you know about invasive fungi that take over native mycorrhizal networks? Have you come across that often?
So there's not been, to my knowledge, a lot of research on that. There is one example, which actually I'll share my screen with you, for instance. So here's a really good example from New Zealand. Like I said, there's not a lot of research on this, so that's not to say this is the only example, but this is just where it was studied and pretty obvious. So there was a single mycorrhizal co-invasive that enabled plant invasion, and in New Zealand there were pine plantations where they were raising pine trees, and the Mycorrhizae without their EMF partners were not invasive. So that's what they're showing you in that 2007 picture. So they might end up getting out of the plantation, but they weren't really invasive. They didn't really take over. And actually the native Pinus cortata is symbiotic with 25 different taxa of Mycorrhizae that we know of. And I'm sorry, it was in Chile, I guess. The trees in Chile never became inoculated intentionally, but somehow became inoculated with an invasive EMF, Suillus luteus, and as a result, that tree then became invasive. So that's what they're showing you in the 2015 picture, is that the tree, basically without its mycorrhizal partner, was not invasive in this new location, but then as soon as it got even just one of its mycorrhizal partners, somehow, we don't know how, it became an invasive species. So not well studied, but yeah, there's some pretty severe consequences if we're not thinking about those things.
Okay, next question. Is there any research about how the type of water, for example, tap well, rain, gray, septic, how that water affects mycorrhizae?
I've never seen a study, so to say that there's not one out there, but I've never come across one. It's something I've also wondered about, since, the chemistry of the water can differ so much based on where it's coming from. But as far as I know, there aren't any studies specifically on that.
And there's a question, if you know, does trichoderma affect growth of the oak wilt fungi?
I don't know. Do you have information that would lead you to think that?
I don't know. That was just the question, and
very specific question. So if there's information on that, I would love to see it.
Yes, I think that's an interesting one. All right, but we're coming up right on 1 o'clock soon, so that's all the time we have for questions. But thank you so much, Lisa. It was a great presentation. Really appreciate it.
Absolutely, my pleasure, and my thanks to everybody joining us today for their interest, and I hope we'll see you out there for future Myco Blitzes and potentially, follow on colonization counts and things like that.
All right, well, thank you so much.